Data organization is the survival skill nobody teaches you. In the next 15 minutes, we will build the habits that make a single-cell or multiome project traceable, auditable, and far easier to defend when a reviewer asks how you got your result. Here is the scenario. 6 months after you submit, reviewer number 2 asks you to reproduce figure 3 with a slightly stricter quality threshold. You open the project folder and find this. None of the files have dates. None of them say what changed. The environment has been updated for another project. Days disappear into reconstructing which script made the figure, and you are on a revision deadline the whole time. This is not a failure of intelligence, and it is not a failure of care. It is a failure of systems, and systems are something you can fix once at the start of a project in about 15 minutes. Disorganized analysis fails in 4 recognizable ways. Script chaos, where nobody can tell which file produced the submitted figure. Version drift, where the clustering was trained on one object and the plots were made from another. Environment decay, where a package update quietly changes your numbers. And collaborator paralysis, where a new student simply cannot run your code at all. Single-cell work is more exposed than most computational biology. The scale is large. One multiome run can produce hundreds of gigabytes. The pipelines are long, with 15 to 25 distinct steps, each carrying parameter choices that propagate downstream. The work is deeply iterative, and papers usually involve several analysts at different institutions. These numbers are illustrative, but the shape of the problem is universal. So here is the plan. 6 habits, a folder structure that gives every file a home, version control that covers code and data together, naming conventions, biological metadata, environment lock files, and notebook hygiene. Each one fixes a specific failure mode. Habit 1, a standardized folder structure. The point is not tidiness. The point is that when every project in the lab has the same layout, the structure itself becomes documentation. A new collaborator, or you in 6 months, can navigate any project without asking a single question. Most templates dump everything into one folder called raw. That conflates categories that behave very differently. BCL and FASC files are the raw sequencing measurement. A cell ranger matrix is not. It already embeds a reference genome, an aligner version, and a set of algorithmic choices. Splitting data into 4 layers tells you instantly what is safe to delete and regenerate, and what is irreplaceable. Here is the full layout. Numbers in the script folders encode pipeline order, so listing the directory shows you the steps in the order they run. Metrics live separately from tables because small JSON metrics belong in Git where they can be diffed. And the logs folder records what a run actually did, the commit, the environment, the random seeds. Now a trap that catches almost everybody. A very common template writes data /raw into gitignore. It looks safe. It quietly breaks everything because DVC puts its pointer files inside those directories. Ignore the parent folder and your data version history never reaches Git at all. Ignore file extensions instead and never ignore the pointer files. One more rule for this section: never hardcode absolute paths. A path pointing at your home directory breaks the moment a collaborator clones the repository. Derive everything from the project root using pathlib. Keep paths in a config file, and keep parameters, thresholds, principal component counts, seeds in params.yaml, so a threshold change does not look like a code change. Habit 2, version control. Without it, your project has no memory. Every time you edit a script, the previous version is gone, and the question what exactly changed between the draft and the submitted analysis becomes pure guesswork. Git was designed for source code, small text files. Commit a 50 GB H5 add, and Git stores the whole object and history on every change. DVC solves this. It keeps the large file in external storage and commits only a small pointer containing the path and a content hash. Git checkout restores your code. DVC checkout restores the matching data. Mechanically, it is simple. You run dvc add on a large file. DVC computes a content hash, writes a tiny pointer file next to the data, and adds the data itself to a local gitignore. You commit the pointer. Your collaborator clones the repository and runs dvc pull and gets exactly the data version that matches the code, verified against the hash. Here is a decision people get wrong. Many tutorials point the DVC remote at cluster scratch. Scratch file systems are usually explicitly temporary, purged on a schedule, excluded from backup. Pointing your only copy of version data there means DVC pull works until the day it does not. Put the remote on durable, backed-up storage. Scratch is an excellent place for the cache, which is meant to be disposable. And credentials. The file.dvc/config is committed to Git, so anything you write there is published to everyone with repository access, permanently, including in history. Use the local flag, which writes to config.local. Better still, use an IAM role or a named SSO profile and store no long-lived keys at all. If a key has ever been committed, rotate it. Removing it from the working tree does not remove it from history. Write commit messages the way you write a lab notebook. Update is useless 6 months later. QC, min genes 500, percent mitochondrial under 10, 82,000 of 95,000 cells pass. That is a complete record of what step ran, with which parameters, and what came out. Add these lines to every job submission script. Months later, when a result is questioned, this is what tells you what ran. The dirty line matters most. A job launch from a working tree with uncommitted changes cannot be tied to any commit, and you want to know that at the time, not afterwards. Restoring a past state becomes a defined procedure. Check out the commit, pull the data, rebuild the environment, rerun the script. One detail worth getting right: use DVC pull, not DVC checkout. Checkout only restores from your local cache. Pull fetches from the remote first, which is what you need on a fresh machine. But be precise about what that gives you. It restores the same inputs, the same code, and an equivalent environment. Whether it reproduces the figure exactly depends on things outside version control, random seeds, thread counts, the linear algebra library, the processor architecture. We will come back to this. Now the part that pays off everything else. Lift every tunable number out of your scripts into a single params file. Thresholds, principal components, neighbors, and the random seed. This is the file a reviewer will ask about and the file you diff when your results move. Then declare the pipeline as a graph. Each stage names its command, its dependencies, the parameters it consumes, and the outputs it produces. Now the relationships between your files are versioned, not just the files themselves. And here is the payoff. Does the conclusion hold a stricter threshold? Change one number in params, run DVC repro, and only the quality control stage, and everything downstream reruns. Nothing else. Then DVC params diff shows exactly what changed, and DVC metrics diff shows exactly what it did to your numbers. The 3-day nightmare from the start of this video becomes a 2-command answer with a table attached. One honest caveat about the lock file. It records content hashes for the declared dependencies and outputs and the concrete values of the declared parameters. That makes it a strong provenance record of what was used. It is not proof that rerunning will produce the same result, and it only sees what you declare. An undeclared input file, or a threshold still hard-coded in a script, will not mark a stage as stale. A quick note on tooling, because people ask. SnakeMake and Nextflow are built for per-sample scattergather across hundreds of FASC files with cluster job submission. DVC pipelines are for the downstream analysis layer, where the value is tying parameters and data versions to Git history. Many labs run both. Habit 3, naming. A file name has exactly one job: to let you identify a file without opening it. Which of the files on the left is most recent? Which one is in the paper? On the right, the date, the content, and the version are explicit, and alphabetical order equals chronological order. The formula has 4 parts. An ISO date, which makes alphabetical order chronological on any file system. A 0 padded step number, so listing a folder shows the run order. A specific snake case description, harmony integration, not integration. And a version label on final figures, which retires the word final forever. One exception. Files produced by a pipeline stage should have stable names, because the stage regenerates the same path each run and DVC already records the date, the hash, and the parameters. Keep the dated convention for scripts, for exported final figures, and for anything a human names by hand. And here is the important limit. It is tempting to pack metadata into file names and split them back out. Do not build a project on that. Metadata changes, and renaming a file breaks everything pointing at it. File names have no schema, no units, no way to say a value is missing. Positional parsing breaks on one extra underscore. And age, sex, and diagnosis in a path leak into logs, error messages, cluster job names, and shared directory listings, places your consent language never anticipated. So the rule is, file names carry stable technical identifiers: donor, specimen, library, run. A version-controlled sample manifest carries the biological, clinical, and batch metadata. You join the two on the identifier, which brings us to the section most projects skip entirely. Habit 4, metadata and provenance. Single-cell data has a nested identity structure: study, donor, specimen, library, sequencing run, and finally the individual cell or nucleus. Conflating these levels is the root of a surprising number of analysis errors. Here is the most expensive version of that mistake. 20,000 cells from 3 donors is a study with an N of 3, not 20,000. Cells from one donor share genotype, handling, dissociation stress, and batch. Treating them as independent replicates inflates significance dramatically. That is why pseudobulk and mixed effects approaches with a donor random effect are now standard. And distinguish clearly between 3 things your manifest should record separately. Biological replicates are different donors, only these increase your N. Technical replicates are different libraries from one specimen, and sequencing runs are the same library across more than one flow cell. They are not interchangeable. The manifest is one small text table, 1 row per library, tracked by Git. Notice the details. Tissue and disease carry ontology identifiers rather than free text, so cortex, and cerebral cortex cannot become 3 groups. Preparation batch and sequencing batch are separate columns because they are different potential confounders and often do not coincide. And a reference identifier links every library to the exact genome build it was aligned against. Now, an important qualification. When you hear track the manifest in Git, that means an approved, pseudonymous projection of it. Stable identifiers and technical fields go in the repository. Restricted clinical attributes stay in controlled access storage governed by your protocol and data use agreement and are joined at analysis time. Commit the schema for every field, including the restricted ones. Analysts then know what exists without the repository holding the values. Next, cell identifiers. A 10x barcode is unique within one library and nowhere else. The whitelist is reused for every run. Merge 2 samples without prefixing and you get silent collisions, 2 unrelated nuclei sharing an index. Prefix with the library. And for paired multiome, note that the gene expression and a C libraries have different library identifiers, so their prefix names never match. Build a shared nucleus identifier from the specimen plus the barcode if you need to relate the 2 modalities. Alignment-derived data is meaningless without knowing what it was aligned to. Record the genome build, the annotation release, the blacklist, the motif database, and a checksum for the reference bundle. Generate file checksums when data arrives and verify them after every transfer. And decide deliberately between H5 add and ZAR. A single file is convenient for exchange, while ZAR suits cloud storage and partial reads. Then turn the data dictionary into something executable. A schema, in Pandora or Link ML or frictionless, is a contract that runs. Note strict equals true. It fails on unexpected columns, which is how an inadvertently added clinical field gets caught before anyone commits it. Put all of this in continuous integration. 5 checks, none of which need cluster access or real data. Scan for secrets across the full history. Rebuild the environment. Validate the manifest and run the tests on a small fixture. Reject large files. And prove that your DVC pointer files are actually visible to Git. That last one catches the gitignore trap from earlier automatically. And a governance box you should read before your first commit if you work with human data. Never commit direct identifiers. Remember that indirect identifiers combine an exact date plus a rare diagnosis plus a single site label can re-identify someone. Genotype and read-level data belong in approved controlled access storage. And a column name check is a smoke alarm, not a de-identification review. That judgment belongs to your IRB or data steward. Habit 5, environments. Take a script you wrote a year ago, build a fresh environment with the latest packages, and run it. Often it fails outright. Sometimes it succeeds with different numbers. 2 researchers running the same script on the same data with different package versions can get genuinely different results through no error of their own. Distinguish an environment file, which is what you asked for, from a lock file, which is what was actually resolved for a specific platform. Use conda export, it is the current command, and it produces multi-platform lock files and CEP23 explicit lock files. And state your versions as last tested on a date, not as permanently current. This tutorial was last tested on the 1st of August, 2026. 3 traps worth knowing. Build strings are tied to the platform you exported from, so a Linux export routinely fails on a collaborator's Mac. A YAML export is a specification, not a lock. The same file can resolve differently later, and mixing conda and pip is where it really breaks down. Current conda actually warns you about this when exporting a lock file, because pip packages are outside what it can lock. Half of single-cell analysis runs in R, and R has the same problem. Use renv, a project private library plus a lock file recording every package version and the R version. Commit the lock file, never the library. One specific warning: ArchR is not a bioconductor package. BIOC Manager install ArchR will not work. Use the lock file the ArchR project publishes, install from GitHub with the bioconductor repositories passed in, or use their official Docker images. If you containerize, know this one. A widely copied pattern activates the environment with shell and then declares an exec form CMD. It does not work, and it fails silently. Shell only applies to RUN and to shell form commands. The JSON array bypasses the shell entirely, so the container runs the base image's Python and every version pin is ignored at runtime. Set the environment on path instead, and then verify it, rather than assuming. And now the honest part. Lock files and containers substantially improve reproducibility. They do not make numerical results deterministic. Random seeds, thread counts, the linear algebra library, the processor architecture, and GPU kernels all still move your numbers. So set seeds from params and log them, pin thread counts, record the architecture, and check that your conclusions are stable under a reasonable perturbation. A finding that survives a seed change is worth far more than one that reproduces to 16 decimal places on one machine. Habit 6. Notebooks. Most single-cell work actually happens in Jupyter, and that is legitimate. Clustering and annotation are interactive, but notebooks fail in 3 ways. Hidden state, where the execution counts read 7, 3, 12, 8, and the displayed result cannot be reproduced by running the file top to bottom. Unreviewable diffs, because a notebook is JSON. And repository bloat, because every rendered figure is stored as base 64 inside the file, and Git keeps every version forever. There are 2 valid policies, and you must pick one. Option A, commit the notebook with output stripped automatically at commit time by nbstripout. Option B, pair the notebook to a plain Python script with Jupytext, commit the script, and gitignore the notebook. A very common misconfiguration installs nbstripout and also gitignores every notebook. The filter then has nothing to act on, and everyone believes they are protected when they are not. Neither tool fixes hidden state. Only one thing does, and it is a habit rather than a package. Restart the kernel and run all cells before you trust a number, before you show a figure at lab meeting, and always before you commit. If it fails, or the numbers move, you have just caught a bug that would otherwise have reached your manuscript. And the graduation rule. Notebooks are for deciding what to do. Scripts are for doing it reproducibly. The moment a notebook produces something that appears in the paper, a filtered object, a differential expression table, a final figure, that logic moves into the scripts folder, takes a dated step-numbered file name, and becomes a stage in the pipeline. The notebook stays as a record of the exploration. It is no longer load-bearing. Put the habits together, and here is what you have. Every figure links back to a script, a parameter set, a data version, a commit, and a recorded environment. A written reanalysis procedure instead of a remembered one. Revisions answered with a parameter change and a diff table. Every cell joining back to a donor, a specimen, and a reference genome. Notebook changes you can actually review, and claims about reproducibility that you can defend precisely. And here they all are in one place, the 10 recurring mistakes. Read them as a checklist against your current project. Most researchers will recognize at least 3, and every one of them is a failure of habit rather than of understanding. Great analysis is not just clever code. It is being able to say, with evidence, exactly how you got here. The complete written tutorial, with all the code, the checklists, and the references, is free at multiomeacademy.com. If this was useful, subscribe and go run git chitignore on your project right now.